So, good evening, students. So, welcome to another session. So, in the previous session, we were started discussing about the mechanism of evolution, right? So, pertaining to the process, or rather the mechanism of evolution, I told you that we'll be studying about two important mechanisms pertaining to the process of evolution. The first one was deviation from Harvey-Weinberg equilibrium, and the second one is different types of evolutionary forces which lead to the process of speciation. So with regards to that, we started off with the first one, which is the deviation from Harvey-Weinberg equilibrium. So in the previous session, we saw what this Harvey-Weinberg equilibrium is all about, right? And we saw what does the Harvey-Weinberg principle state also. So just a quick relook of what we have spoken in the previous session, right? So what did we say regarding the Harvey-Weinberg principle? We said that the Harvey-Weinberg principle states that the allele frequency in a particular population would remain constant from generation to generation under certain conditions. So I gave you the list of those conditions also. Under what conditions the allele frequency or the genotypic frequency would be constant. So what are those conditions what we saw in the previous session? The first one, the population size should be large. Second one, there should be random mating in the population, right? The third one, what we saw was all of these organisms, right, should the second one, that is, there should be random mating in that particular population. And the third one, what we actually said was, right, a few more topics we have already discussed with regards to that, right? So there should be no differential reproductive success in that particular population. All the organisms should be of the homogeneous age, right? So these are the conditions. There should be no evolutionary forces acting upon that particular population, right? So these are the 7 different conditions what we saw pertaining to the Harvey-Weinberg equilibrium, right? So if all of those conditions are maintained, then the Harvey-Weinberg equilibrium will be established. That is, the allele frequency would remain constant from generation to generation. Then we also spoke about the significance of Harvey-Weinberg principle. So we also saw what this Harvey-Weinberg principle states, or what does it conclude, right? So we saw that any deviation in one or more than one of these conditions, what we saw, right, would lead to disturbance in the equilibrium, that is genetic equilibrium, and that disturbance would lead to some sort of changes in either the allele frequency or the genotypic frequency, or both. And this change in the allele frequency would bring about some sort of variations in that particular organism, and these variations would now lead to the process of speciation, that is formation of a new species, or rather evolution of a new species. That's the reason why we call variations as the raw materials for the process of evolution, right? So in continuation with that, we also took up something called as the Harvey-Weinberg equation, right? So we said that it is a mathematical model to explain genetic equilibrium, right? A mathematical model to explain genetic equilibrium. And we saw those two equations also. So if you could recollect what are those two equations what we saw, we said that P plus Q would be equal to 1. So this is the first expression what we saw, P plus Q equal to 1. The second one, P square plus 2PQ plus Q square would be again equal to 1. So this would be termed as the allele frequency, and this would be termed as the genotypic frequency. So we said that the allele frequency or the genotypic frequency would remain constant from generation to generation under those 5 different conditions what we saw in the previous session, right? Now in this, what does P represent? P would represent the allele frequency of a dominant allele. Q would represent frequency of a recessive allele. P square would represent the frequency of a homozygous dominant genotype. 2PQ would represent the frequency of a heterozygous dominant genotype, and Q square would represent the frequency of the recessive genotype. Right? So these are the two expressions what I told you to remember in the previous session. Right? So in today's session, what we'll be taking up is, we'll take up a couple of examples with regards to this particular Harvey-Weinberg equation, because several times in your exam, a lot of questions have been asked based on this particular topic, that is Harvey-Weinberg equilibrium. So you need to know how to solve those problems, right? So there are 2 example problems given in our books, right? So if you have your material, right, that is our college material, so you can just open that right? So you can just open it to page number 82 in that material, right? So it is part B, right? So volume 2, part B. So page number 82, we have two example problems given in our books. So we'll try solving these two example problems, and once we are done with that, in our exercises again, there are some more problems given in our exercises. So you can start working out on those exercises based on this particular model, right? Now let's take up the first two problems which are there, or two simple problems which are there in page number 82 and 83, right? So let's begin with the first one. The first problem what we'll be taking up is, right? So the first problem what we'll be taking up is, it says that assume that in a population of 600 individuals, right? Assume that in a population of 600 individuals, 256 individuals are recessive, right? So the recessive individuals are said to be 256, right? So in a population of 1600 individuals, the recessive individuals are said to be 256. Now they are telling us to find out homozygous dominant and heterozygous individuals in the population, right? So find out homozygous dominant as well as the heterozygous dominant individuals in that particular population, right? So they are telling us to find out homozygous dominant as well as the heterozygous dominant individuals in the population. So this is the question that is being given. So what does the question say? In a population of 1600 individuals, the recessive individuals are said to be 256 recessive individuals. Now from this data what they have given us, we need to find out how many of the individuals are said to be homozygous dominant and how many individuals are said to be heterozygous dominant, right? So for this, what we need to do, right? So what we need to do for calculating this. Now, based on the data what we have given, what is being given, we have the number of recessive individuals to be 256 individuals, right? Now, when we say recessive, we have these two formulas to remember, or these two expressions to remember. So all our problems, whatever we get with regards to Harvey-Weinberg principle, will be based on these two itself. So you need to know when to use P plus Q equals 1. At the same time, you need to know when to use P square plus 2PQ plus Q square equals 1. One hint let me give you. Whenever any sort of problem with regards to Harvey-Weinberg principle is given, whatever be the question, first try to find out Q square as well as Q, right? So this is the hint what you need to know, right? So whichever problem it is, first try to find out Q square as well as Q. So if you're able to find out Q, right, you can find out P from the first expression. Once you find out P and Q, we can find out 2PQ also. Now let's get back to this. First one, what they have given is recessive individuals, that is 256. So when we say recessive individuals, we are talking about the recessive genotypes, right? We are talking about the recessive genotypes and not the recessive alleles. We are talking about the recessive genotypes. Now, from this data what we have, that is 256 recessive individuals, let us first find out the recessive genotypic frequency. Right? Let us find out the recessive phenotypic frequency. So how do we find out the recessive phenotypic frequency? Sorry, genotypic frequency that represents Q square. So this is our recessive genotypic frequency. Now how do we find out the recessive frequency or recessive genotypic frequency? Total number of recessive individuals 256 divided by the total number of population, that is 1600. Right? So 256 by 1600 would give us the frequency of the recessive genotype. This will come to approximately 0.16. So this is our Q square. So the recessive genotypic frequency is said to be 0.16. Now since we have got Q square, we can easily get Q. So what would be Q here? Q would be root of 0.16. Right? So how much is root of 0.16? It would be 0.4. Now this is our Q value. Now we have got Q square and we have got Q also. Right? So our Q value is 0.4 and Q square would be 0.16. Now, since we have got our Q value, apply this P plus Q equals 1, so that we'll be getting the P value, right? So what would be the P value here now? So the P value would be, we know that P plus Q equals 1. Now from this, since we have Q value, so P would be 1 minus Q, that is 1 minus 0.4. So 1 minus 0.4, it would be 0.6. So this is our P value, right? So the P value is said to be 0.6. That is, the frequency of the dominant allele is 0.6. Frequency of recessive allele is said to be 0.4. Now, since we have got P, what they have asked in the question, they have told to find out homozygous dominant individuals. Now, when we say homozygous dominant individuals, we are actually talking about P square. So this would actually be P square and this would be 2PQ. So we need to find out how many are said to be homozygous dominant, how many are said to be heterozygous dominant. Now, since we have already got P value, getting P square from this is very simple, right? So P square would be how much? So our P square value would be 0.6. P is 0.6. So this would be 0.6 into 0.6. It would be 0.36. So our P square value is said to be 0.36. Right? So this is our P square value. Now, since we have got P square, now what we need to find out is we have got the frequency of the dominant genotype. Now, what they have asked in the question, if they ask you to find out what is the frequency of dominant genotype, we would stop the answer here itself. But in the question, what they have asked is, they told us to find out the number of homozygous dominant individuals. So how do we get the number of dominant individuals? Number of dominant individuals here would be 0.36, that is the frequency, into the total number of population. The total number of individuals present in the population would give us the homozygous dominant individual. So how much would this come to? 1600 into 0.6, it would come to approximately 576, right? So it would be approximately 576. So this is our answer here. So homozygous dominant individuals in the population would be 576. Yeah, right? Similarly, they told us to find out heterozygous dominant individuals. That is, we need to find out 2PQ. So to get 2PQ, what we need to do? 2PQ, we have P and we have Q. So this would be 2 into our P value would be 0.6 and our Q value would be 0.4. So 2 into 0.6 into 0.4. So that would give us almost 0.48. 0.48. So this is the frequency of the heterozygous dominant genotype. Now, from the frequency of heterozygous dominant genotype, if we want to get the number of heterozygous dominant individuals, so this would be 0.48 into the total number of individuals in the population, that is 1600. So how much would this come to? It would come to almost 768. So there are 768 individuals, and these 768 individuals are said to be heterozygous dominant individuals. 768. So this is how we actually start doing it, right? So what they have given here in the question is, they have given the population size, the size of the population, right? So the size of the population is said to be 1600, right? And they have given us the data with regards to the recessive individuals in the population. So the recessive individuals in the population is said to be 256. Now from this data, they told us to find out the homozygous dominant individuals. How many are said to be homozygous dominant and how many are said to be heterozygous dominant individuals. So for that, what we need to do is, based on the data what we have got, first find out the frequency of the recessive genotype. Since what they have given us is recessive individuals, which represents the recessive genotype. Just remember this. Whenever they have given you a question based on recessive individual, that would represent recessive genotypes. First step, what you need to do is first find out the frequency of the recessive genotype, Q square. So how do we get Q square? Total number of recessive individuals divided by the total number of population. So that would give us almost 0.16. So this is the frequency of the recessive genotype. From this frequency, we are now calculating the frequency of the recessive allele. Right? We are calculating the frequency of the recessive allele. Right? So the frequency of the recessive allele now would be 0.4. As I've already said at the beginning, whatever be the question, first try to find out Q square, then get your Q. Once you get the frequency of the recessive allele, getting the frequency of the dominant allele is very simple. By applying this particular expression, P plus Q equals 1. Right? By this, P would be 1 minus Q, that is 0.4, and the frequency of the dominant allele would be 0.6. From the frequency of the dominant allele, we start calculating the frequency of the homozygous dominant genotype, 0.36. From this, total number of homozygous dominant individuals in the population, 0.36, that is frequency into total number of individuals in the population. So that would give us 576. So among these 1600 individuals, right, among the 1600 individuals, the total number of homozygous dominant individuals would be 576. From this, again, calculate heterozygous dominant individuals. For that, first we start calculating the heterozygous dominant genotypic frequency, that is 2pq. Now we have P and we have Q. Start substituting those values. So 2 into 0.6 into 0.4 would give us 0.48. Total number of individuals, 1600. So if we multiply the frequency with the total number of individuals, we'll be getting the total number of heterozygous dominant individuals in a population. Right? Still, if you want to revalidate your results, right, what expressions we have is P plus Q is equal to 1. So start calculating P plus Q1. Are you getting 1? If you're getting 1, the answer is correct. Second one, P square plus 2PQ plus Q square also should be equal to 1. So our P square here is 0.36, Q square is 0.16, and 2PQ is 0.48. Now when you add all of these three values, we should get 1. Right? So that's how we start revalidating whether we are getting the correct results or not. One more way, start calculating all of these individuals present in the population. Number of recessive individuals is 256, homozygous dominant 576, heterozygous dominant 768. So when you add up all of these three populations, are you getting 1600? Right? So that's how we actually start revalidating our results, whether we are doing it properly or not. Right? So this is one simple problem what we have with regards to Hardy-Weinberg equilibrium. So most of the questions what you'll be getting from this topic are all problem-based questions. It would be more or less similar to this one. But you need to know when you need to use P plus Q equals 1, at the same time when you need to use P square plus 2PQ plus Q square equals 1. Right? So this is the first problem what we have with regards to the Hardy-Weinberg principle. Right? Hope all are clear with this one. Right? So if you're clear with this, we'll proceed with the next example what we have in our books. So problem number 2, example problem number 2. So let's take up with the example problem number 2. This problem is slightly different, right? Slightly different, but you can just remember this also. Sometimes these sort of questions also will be given, right? Slightly deviating. Okay, so let's write down the question here. The first one, what they say is, assume that in a population, again, they have given you the population. Assume that in a population of 200 individuals, right? The population size is said to be 200 here. The second thing what they have given is, out of 200 individuals, homozygous dominant individuals is said to be 114, 114. Then heterozygous dominant are individuals are said to be 76, and recessive individuals are said to be recessive individuals are said to be 10. So this is the data what they have given. So the population size is said to be 200 individuals. Out of 200 individuals, the homozygous dominant are said to be 114, heterozygous dominant are said to be 76, and the recessive individuals are said to be 10. So from this data, what they are telling us to calculate is, find out the dominant and recessive allele frequency, right? So they are telling us to find out the dominant allele frequency, right? So they are telling us to find out the dominant allele frequency, at the same time the recessive allele frequency, the dominant allelic frequency as well as the recessive allele frequency. So this is what they are telling us to find out. So what they have given us is the number of individuals in homozygous dominant, heterozygous dominant, and the recessive individuals. From this, they are telling us to find out the dominant allele frequency and the recessive allele frequency. What I told you in the previous session, in the previous problem, when they tell you about homozygous dominant individuals, they are representing about, this would be Q square. So they are representing about, this would be P square. Heterozygous dominant individuals would represent 2PQ. Recessive individuals would represent Q square. Now what they are telling us to find out is dominant allele frequency. That is, they are telling us to find out P and Q. Right? So giving P square, Q square and 2PQ, they are telling us to find out P and Q. So if this sort of a problem is given, we cannot use any of these two expressions directly, right? So we can say that since P square is given 114, from P square directly we can take it as P, right? You can say it like that, but that should not be done. Why it should not be done? Because in case of dominant individuals, when we talk about P here, dominant allele, P dominant allele is present in homozygous dominant as well as heterozygous dominant also. So we need to find out the dominant allele frequency in homozygous dominant individuals. Similarly, we need to find out the dominant allele frequency in heterozygous dominant individuals also. Similar case with Q also. So when we talk about recessive frequency, recessive allele frequency, recessive alleles are present in homozygous recessive condition also, and at the same time recessive alleles are present in heterozygous conditions also. So that is the case, directly we cannot calculate P or Q from P square and Q square, because P and Q is present even in 2PQ conditions also. Right. So if this sort of a problem is given, right? If this sort of a problem is given, there is a simple expression again what we use. A simple expression again what we use. So let me put that down. So the simple expression. So first let us take up with dominant allele frequency. So when we have to calculate the dominant allele frequency P, right? So the expression what we use for this is, right? So the expression what we use for this is 2 into number of homozygous dominant individuals, right? 2 into number of homozygous dominant individuals plus the number of heterozygous dominant individuals, divided by 2 into total population, right? So this is the simple expression what we use. Whenever we are calculating for dominant allele frequency, that is P, we use the expression 2 into total number of homozygous dominant individuals plus the number of heterozygous individuals divided by the total population size. So when we start using this particular expression, let us see what we'll be getting. Right? Let's see what we'll be getting. So let's start substituting the values here. Right? So for P, so it would be 2 into number of dominant individuals is said to be 114 plus number of heterozygous individuals is said to be 76 divided by 2 into total population is said to be 200. 2 into 200. Right? So it would be 2 into 114 plus 76 divided by 2 into 200. So how much do we get for this? So it would come to approximately 304 divided by 400. 304 divided by 400. So this would give us 0.76, right? So this is the frequency of the dominant allele. This would be the frequency of the dominant allele, 0.76, right? So how did we arrive to this particular value? 2 into number of homozygous dominant individuals. Homozygous dominant individuals are said to be 14, right? Next, plus the number of heterozygous dominant individuals. Heterozygous dominant individuals are said to be 76, divided by 2 into total population. So 2 into total population, 2 into 200. So when we start substituting the values, 2 into 114, so this will be 228, 228 plus 76, right? 228 plus 76, that would give us 304 divided by 400. So when we start simplifying this, we get 0.76. So our p value here would be how much? It would be 0.76. So this is the dominant allele frequency what we have, which is said to be 0.76. Similarly, if we have to do it for Q also, right? Similarly for Q also, if we have to do it, right? So Q also we start using the same expression here, but slightly different here. Since Q represents recessive individuals, right? We'll be slightly modifying this. Modification will be only in the first part. So this would be 2 into number of recessive individuals, right? It would be 2 into number of recessive individuals, 2 into number of recessive individuals plus number of heterozygous dominant individuals, right? Divided by 2 into total number of population. So 2 into total population present in that particular area, right? So when we start substituting it again, so it would be 2 into number of recessive individuals are said to be 10 plus number of heterozygous dominant is said to be 76 divided by 2 into total number of population, that will be 2 into 200. So when we start simplifying this, how much do we get? On here we will be getting 96 divided by 400. 96 divided by 400. So when we start simplifying this, the value what we'll be getting is, it will be 0.24. 0.24. So this would be our recessive allele frequency. So the recessive allele frequency would be 0.24, 0.24. In simple, one more way also what we can do is, so this would be 0.76 and this would be 0.24, right? One more way also what we can do is, once you get your p value, we can find out q also from p plus q equals 1. Anyways, that way also you will be getting 0.2. But this would be the correct method how we actually start doing, right? So this is what is we have with the second model with regards to the Harvey-Weinberg principle. So in this model, what we saw is the total population is given. Along with that, the number of homozygous dominant individuals as well as the number of heterozygous dominant individuals are also given. From this, if they tell you to find out the dominant allele frequency as well as the recessive allele frequency, that is, as I told you, if homozygous dominant individuals would represent the p square, that is genotypic frequency, and heterozygous dominant individuals would represent 2pq, and recessive individuals would represent q square. So from this, if they tell you to find out p and q, that is the dominant allele frequency as well as the recessive allele frequency, right? So how do we do it? We use a simple expression. So if you have to find out the dominant allele frequency here, so the dominant allele frequency p would be 2 into number of homozygous dominant individuals plus the number of heterozygous dominant divided by total population. So when you start simplifying this, we'll be getting 0.76. So 0.76 would be our dominant allelic frequency. Similarly, if you have to find out Q, right? So this would be Q here. So if you have to find out Q, so recessive allelic frequency would be 2 into number of recessive individuals in the population plus number of heterozygous dominant divided by 2 into total population. So when we start substituting the values, what we get? So our Q value would come to 0.2. So the value is 0.2. So if you want to still extend this, again you can find out P square, right, from this value. You can find out Q square from this value. again you can start finding out 2pq also, right? So you can again revalidate your results by calculating p square, q square and 2pq, again multiply it with the population and see whether you're getting 114, 76 and 10s respectively here, right? So this is what we have with the second type of problems what we have. So two example problems we have taken up, right, in this session, right? So you can just start going through other problems also which are there in your exercises, right? So in your exercises still some more problems are there, more or less with the same model, right? So you can start practicing those questions also which are there in your exercises, because as I've already said, most of the questions from this topic will be all problematic questions, almost similar to this sort of things what we have seen, right? I hope all are clear with this particular concept, the Harvey-Weinberg principle. So you can quickly make a note of it, right? So once you're done with this, we'll move on to the second mechanism of evolution, right? So we are still in the first mechanism of evolution, which is called the deviation from Harvey-Weinberg equilibrium. So what we have seen so far is any sort of deviation from Harvey-Weinberg equilibrium, right, would lead to formation of variations in an organisms, and that variations would lead to the process of speciation, right? So what we are telling here is in all of these conditions there is no speciation happening. Why there is no speciation happening? Because p plus q is always equal to 1, p square plus 2pq plus q square is always equal to 1. So what does the Harvey-Weinberg equilibrium state again? The allele frequency or the genotypic frequency in a population would always remain constant under certain conditions from generation to generation. So that is what we have actually taken. So any deviation in one or more than one of the conditions what we have seen will disturb this equilibrium, right? So once this equilibrium is disturbed, it would lead to some sort of changes in the allele frequency or genotypic frequency, leading to variations in a particular organism. And those variations would get accumulated, and over a period of time, it would lead to reproductive isolation, and that reproductive isolation would now lead to the evolution of a new species. The first mechanism of evolution, what we are studying, which is called as the Harvey-Weinberg equilibrium. Right? I hope this concept is clear. Right? Now let's move on into the second mechanism what we have. Right? The second mechanism what we'll be taking up is the evolutionary forces. Right? So let's take up with the evolutionary forces. So what are these evolutionary forces? So if you could remember when we were discussing Harvey-Weinberg equilibrium also, some of the conditions which maintains Harvey-Weinberg equilibrium, we said that there should be no evolutionary forces acting on that particular population. That's what we have said, right? We said that there should be no evolutionary forces acting upon that particular population. So what we are dealing here is now evolutionary forces. Evolutionary forces. So what are these evolutionary forces all about, right? Any forces or factors, right? Try to understand this. Any forces or factors which would lead to the change in the gene pool of a population, right? So let me repeat it once again. What is evolutionary forces? Any force, any sort of force that would lead to change in the gene pool of a population is called as an evolutionary force, right? So what do we mean by gene pool? Yesterday in the previous session we saw about gene pool also. So what is the gene pool? The sum total of all the genes present in a sexually reproducing population at a given time period is called as gene pool. Right. So what are these evolutionary forces? Any forces or factors which brings about the changes in the gene pool of a population is what we call it as the evolutionary forces. Right. And these evolutionary forces are the ones which are responsible for the process of speciation, that is for the evolution of a new species. Right. So there are various evolutionary forces which are being present. So we'll see what are those evolutionary forces. So first I'll just give you the list of these evolutionary forces. Then we'll be discussing each of the evolutionary forces in detail. So the first evolutionary forces which brings about the change in the gene pool is natural selection. Right? It is natural selection. So this is the first evolutionary force which brings about changes in the gene pool, right? It's called as the natural selection. The second evolutionary force, what we'll be talking about, is called as gene flow, right? It is called as gene flow, or it is also called as gene migration, right? Gene flow or gene migration, right? So this is another evolutionary force which brings about some sort of changes in the gene pool. The third evolutionary force, what we'll be talking about, is called as the genetic load, right? The third evolutionary force is the genetic load. The fourth evolutionary force, what we'll be talking about, is called as genetic drift. So this is another evolutionary force which is called as genetic drift. And number 5, what we'll be talking about is what we have already studied. It is called mutation. So we have studied a lot about mutation. So this mutation is also considered as an evolutionary force. And the last evolutionary force also we have already studied, which is called as genetic recombination. Genetic recombination. So we have already studied the last two terms. So we are not going to get into much details with regards to the last two terms, that is mutation as well as genetic recombination. But we'll be taking up each of these particular forces in detail. The first one is natural selection. The second one is gene flow or gene migration. The third one is genetic load. The fourth one is genetic drift. Number 5, mutation. And number 6, genetic recombination. Right? So these are the 6 forces what we'll be studying. And these 6 forces or factors are actually responsible for bringing about some sort of changes in the gene pool. Right? So what did we say with regards to these evolutionary forces? So what are evolutionary forces? Any forces or factors which brings about some sort of changes in the gene pool of a particular population is this evolutionary force. Some of the major evolutionary forces what we'll be studying is the natural selection, the gene flow or the gene migration, genetic load, genetic drift, mutation, as well as genetic recombination. Right? So let's begin with the first one, which is called as the natural selection. Right? So let's take up with the first one, natural selection. So about natural selection also, we have already studied a lot about natural selection, right? Because when we spoke about Darwin's theory of evolution, we spoke in detail with regards to the process of natural selection. So that's the reason why I'm not going to get into much details with regards to natural selection. So what we'll see here is, right, what we'll see here is there are different types of natural selections which are being present. So directly we'll get into different types of natural selection, right? So there are 3 different types of natural selections which are being identified, right? 3 different types of natural selection. The first type of natural selection is called as stabilizing selection. Stabilizing selection. So this is the first type of selection, natural selection, which is called as stabilizing selection. Now this stabilizing selection is also called as centripetal selection, right? It is also called as centripetal selection. Either stabilizing selection or centripetal selection. The second type of selection what we'll be studying is called as directional selection. Second one is directional selection. So this is the second type of selection what we'll be taking up. The third type of selection what we'll be taking up is called as disruptive. The third one is called disruptive selection. And this disruptive selection is also called as centrifugal selection. Right? So these are the 3 different types of selections what we'll be studying under one of the major evolutionary forces, which is called as natural selection. Stabilizing selection, also called as the centripetal selection. Second one is directional selection. And the third one what we'll be studying is called as the disruptive selection. Right? Stabilizing selection, directional selection, as well as disruptive selection. So let us quickly begin with the first one, which is called as stabilizing selection. Right? So let us take up with the stabilizing selection, then we'll come back with these 2 types of selection. Now, what is this stabilizing selection all about? right? So what is the stabilizing selection all about? Now, things what you need to remember here is when does this type of selection operates, right? So what you need to know is when does each of the type of selection operate, right? So I'll tell you that, so you can just make a note of that also. So this type of selection, that is the stabilizing selection, usually operates at stable environmental conditions, right? So they usually operate in stable environmental conditions. So what does this mean? Wherever the environmental conditions remains unchanged, wherever there is constant environmental conditions, only there this sort of selection called stabilizing selection will operate. Right? So the first and foremost thing what you need to remember is this type of selection called the stabilizing selection will be operated only in the condition where there is stable environmental condition. Right? This is the first one what you need to remember. Now, what exactly happens in this type of selection? What happens in this type of selection? One thing what you need to remember here is this curve, we usually call it as the phenotypic distribution curve. Right? This is the phenotypic distribution curve. We usually call it as the phenotypic distribution curve. Now, what happens in stabilizing selection here is, right? These are the organisms that is present here in the peak region. Right? So the organisms that is present in the peak region are said to be the fittest organisms. Right? So what you need to remember here is organisms present in the peak, that is this particular region. Right? Organisms present, say, in this particular region, they are said to be the fittest organism. So we start calling them as organisms showing average phenotype, right? So this is the place where the organisms show average phenotype. So we, in other terms, we call them as the fittest organisms. Right. Now, what exactly is happening in this type of selection is organisms present in the this particular region, that is the peak region, are selected by the nature, right? So organisms present here alone are selected by the nature, whereas the organisms present towards the both the extremes of the phenotypic distribution curve are said to be eliminated. They are not selected by the nature. This type of selection, we start calling it as stabilizing selection. Right? So what happens in stabilizing selection? First thing what you need to remember, stabilizing selection will operate only in stable environmental condition. Right? Number one, stabilizing selection always operates only in the stable environmental condition. Second important thing what you need to remember here is, in this type of selection, organisms with average phenotype, that is, the fittest organisms are preserved, or rather selected by the nature, whereas individuals present on both the extremes are eliminated by the nature. Right? Individuals present on both the extremes of this extreme and this extreme of the phenotypic distribution curve are said to be eliminated by the nature, while the organisms present in the middle part, that is the peak region, what we call it as organisms showing average phenotype, they alone are selected by the nature. So this curve, which was like that, after the process of selection, would become somewhat like this, right? It would become somewhat like this, right? Why it would become somewhat like this? Because the individuals that are present in both the extremes are being eliminated, as a result of which the individuals possessing the average phenotype are alone selected by the nature. Clear this? This type of selection we start calling it as stabilizing selection. Now, one more thing what you need to remember with regards to stabilizing selection here is, right, since organisms which are said to be fit alone are being selected by the nature, there would not be any evolutionary changes present in this particular population. Right? So, things what you need to remember here is there would be no evolutionary changes, in the population. Why there would be no evolutionary changes? Because only the stable organisms or stable individuals or the fit individuals alone are being selected. But it maintains, right? What it maintains? It maintains phenotypic stability. It would maintain phenotypic stability from generation to generation. This is the characteristic feature of this particular type of selection, which we call it as the stabilizing selection, right? So what happens in stabilizing selection is, since organisms present towards the center of the phenotypic distribution curve, that is having the peak value, that is organisms which are said to be fit, since they alone are being selected by the nature, right? Since they alone are selected by the nature, there would be no evolutionary changes in that particular population. Why? Because the organisms present in the peak region, organisms showing average phenotype, are alone selected by the nature, right? But what it does is it maintains the phenotypic stability from generation to generation, right? So that is the characteristic feature of this sort of selection, what we call it as the stabilizing selection, right? So quickly going through the stabilizing selection once again. What is the stabilizing selection? When does it operate? It usually operates only in a stable environmental conditions, right? It would operate only in a stable environmental condition. Now, what happens in this sort of selection? Organisms showing average phenotype, they alone are preserved or selected by the nature, while the organisms present towards the both the extreme ends of the phenotypic distribution are eliminated by the nature. As a result of which, what would happen? Only the organisms which are stable, showing average phenotypic characters or average phenotype, are alone selected by the nature. Since only the organisms or individuals exhibiting average phenotype are selected by the nature, there would be no evolutionary changes pertaining to the process of speciation in that particular population. But this type of selection would maintain phenotypic stability from generation to generation, right? So this is what we call it as the stabilizing selection, or it is also called as the centripetal selection, right? So this is the first type of natural selection what we have, which is called as the stabilizing selection as well as the centripetal selection, right? So once we are done with this, we have two more selection processes, which is called as directional selection, and the other one is called the disruptive selection. So those two different types of selection, we'll take it up in the next session.